A very warm welcome to this webinar this afternoon. My name is Ingrid Brands. I'm the CEO of BioStrand. And before I introduce my guests of the Data Science Institute of the University of Hasselt, I will first make some very practical remarks. If you have any questions during the presentation, just post them in the Q&A box. We will pick them up. We will answer them during the Q&A session, which is at the end of the webinar. If we cannot answer your question at that time, we will come back to you by email. So I'm really very glad to introduce you to Professor Valkenborg of the Data Science Institute of the University of Hasselt. He's really an expert in the field of bioinformatics and proteomics. And we asked Professor Valkenborg to do a validation study on the technology behind BioStrand's Retrieve and Relate. And today he's going to present the results of the benchmark study that was performed to compare sequence retrieval between BioStrand's RNR and BLAST. And our other guest today is Dr. Joris Van Houtven. He conducted his PhD research under the supervision of Professor Valkenborg and successfully defended his PhD thesis last month. And his work was about finding a way to optimize mass spectrometry data analysis. And although at first sight, the two topics might seem very different, there is a very interesting linking pin between the two presentations. And the linking pin, the central theme of the webinar is patterns. And why patterns? Well, BioStrand has developed a completely new methodology for omics data analysis in which we use patterns to do all the work and to perform all the tasks in bioinformatics. And then in the research of Joris Van Houtven, patterns play a key role in designing a very exhaustive and comprehensive way of searching through and analyzing the mass spectrometry data. Before we go to the talk of Professor Valkenborg, I would like to give you a quick overview of Retrieve and Relate because it's a relatively new tool. We launched it at the end of last year. And Retrieve and Relate is our very powerful search tool. What can you do with it? Well, it's meant to support sequence retrieval. You can search for similar sequences. You can search for functions, annotations, do alignments. And our goal was not only to develop a search tool, but really to develop a next generation search engine that is beneath it. A next generation search engine that is really able to operate in a big data environment. A next generation search engine in which the data are really integrated. And at the same time, the tool has to be very convenient. So ultimately, if you use Retrieve and Relate, what do you get? Well, with Retrieve and Relate, you can search through 440 million reference sequences that reside in the 11 most popular multi-omics databases. But you even can search through the sequences that are in patent databases. And this search is conducted simultaneously. On top of that, you can add your own proprietary data to the search base. Of course, it's always securely stored. And despite the size of the data, the searches are very fast. So on average, any search is conducted under 3 seconds. Then what you also get is an integration of the omics layers. And it means that you always search simultaneously through all the omics layers, through DNA, RNA, and amino acids simultaneously. So you paste your sequence in, and we give you automatically all the translations. It's convenient. As you can see on the right side of the slide, we only have one search bar. There you put in all your searches, whether it is DNA, RNA, or amino acids. And you can also search by text, by free text. No parameters to set. We have aggregated all the metadata, so you first always get a very comprehensive overview, and then you decide how to drill down to the details, and you can go back and forth replicate your search or do other things. So that's a little bit an overview of what RNR does. RNR is the search tool to retrieve the similar sequences. It's new technology, and that's why we asked Professor Valkenborg to validate the search engine that's underneath, the algorithm that is underneath retrieve and relate. And yeah, when you evaluate search engines, you can do that using multiple criteria. You can look at speed, you can look at the magnitude of the search space, you can look at the level of integration. But in this validation study, we only focused on the relevance of the retrieved results. And what does that mean? How relevant are the results to the user's intent? Is what you find related to your search, and is it as complete as possible? But for this purpose, then we compare retrieve and relate against the BLAST algorithm, which is a reference tool in bioinformatics, in performing then a classification task. And the CAT classification provides then a suitable notion of the ground truth for such a classification task, even though CAT and PDB databases are relatively small. So that's a bit the scene, the introduction. And now, Dirk, the floor is yours to tell the results of that validation study.
Okay, I will first unmute, so I hope I'm loud and clear, and I will now share my screen. So I hope that went okay. So if everything is correct, you should see my screen and hear my voice. Is that okay? Yes?
Okay. Yes, all set.
Okay, well, first of all, Ingrid, I want to thank you for this very nice introduction. And also I want to thank BioStrand and the entire crew for the opportunity to present a part of the work we did together here at this seminar. So as Ingrid already introduced, my name is Dirk Valkenborg, and I'm a professor of bioinformatics and data science at the Center for Statistics here at Hasselt University, and also a member of the Data Science Institute, also at Hasselt University. I will not do a lengthy introduction about these institutes. If you want to learn more about our research lines, or you want to learn about how we were involved in COVID-19 research in Belgium, please visit our websites displayed here at these links, or just send me a direct message or contact Sarah because she is our research manager at the Data Science Institute. Okay, so an important problem in bioinformatics and computational biology is the following. Let's say I have a biological sequence, and it can be any type of sequence. It can be DNA, RNA, or a polypeptide chain composed of 20 amino acids. And you want to know a little bit more about that unknown sequence that you have. So what you will do, or what you can do, is go out into the world and try to figure out what other scientists have discovered about related sequences. So an important mission here is to, well, relate the sequence that you care about and relate that to the entire ecosystem of data, sequence databases that is out there. And that's a sensible thing to do because we know all about evolution. We know about homologous genes and proteins. It means it are genes and proteins that we inherit from a common ancestor. And when there is information about these homologous genes and proteins, we could infer maybe also some functionality about that for our own precious sequence that we have at our hand here. So biological sequence retrieval, that is here the name of the game. And unfortunately, that sounds a little bit easier than that. Let's say I'm a computer scientist and I might think, easy peasy. I know a great deal about databases and I know how to structure large data repositories. So in computer science, we will pre-index the database. Or something like we organize our data and keep an index of where every piece of information exactly is sitting in that database. And if we then have the sequence that we're interested in, Well, that sequence is a string. It's a string of 4 letters if it's a nucleotide, or 20 different letters if it is an amino acid. So I can go with that string and query my index database and quickly retrieve all the words that contain my string. So, problem solved. Unfortunately, that's not the case, because biology has to do a lot of things to copy our precious DNA. So, for example, it needs to unwind, it needs to provide energy, provide sufficient nucleotides. So a lot of things can go wrong, and we call that mutation event. So biology will make mistakes. And there are a lot of mistakes here displayed on this slide. I will not go over them, but the issue here is that if you want to do this search by indexing, pre-indexing databases, we need to become error tolerant. And so the whole indexing idea is not so appealing anymore, because suddenly we need to consider a lot of variants and synonyms of the string. So that is an issue. So how did bioinformatics solve that issue? Well, let's have a look. So in the 70s, people were thinking like, okay, I need to become error tolerant. I need to find similar sequences, taking into account the mutational events that an errors biology will introduce. So I need to come up with an algorithm that maximizes the similarity between 2 sequences. That is more or less that maximizing is the key idea about the first global alignment algorithm invented in the 70s by Needleman Wunsch. And that is a dynamic programming algorithm. It is pretty fast. It's faster than brute force, trying to find all possible optimal alignments. But it's actually not fit for purpose anymore today, because we have too much genetic and information, and also information about proteins. So let's move on a decade. We go to the 80s. Then Smith-Waterman came up with a local alignment algorithm, also very popular in that day. It was also based on dynamic programming and also optimal algorithm. So it would find optimal similarity between these 2 sequences, and then we could evaluate if that similarity is sufficient to relate these 2 protein sequences, for example. But again, it's a little bit slow. So in the 19s, we have an uptake of genomic information. Databases grow bigger. These algorithms were no longer sufficient. So a bunch of heuristic algorithms were invented that mimic the local alignment. So that's to name a few, FASTA or BLAST. And what these algorithms did was they interchange the optimality to find similar sequences with speed. So these ones were faster. And then from 90s on, we moved to the year 2000 up to this stage actually. Some improvement was done on the algorithm, but most of the speed up that was achieved could be attributed to technological innovations. For example, like parallel and distributed computing, or the rise of GPUs and cloud computing, that all contributed to a scale up of these heuristic algorithms. But still, it is not an optimal algorithm, and thanks to these technological innovation, it is a bit faster than the algorithm from the 90s. But still, we're confronted with a very fast expansion of genetic and other omics data, and that challenges the speed and scalability of current sequence alignment algorithms, and impose a need also for better data integration, because we want to perhaps also now combine information from not only genomic information, but also want to link genomic information to information from epitope databases and so forth. So the landscape becomes not only more voluminous, but also more diverse. And that is an issue. So Then, well, to recapitulate, up till now, sequence alignment and sequence similarity, those were the things that you had to do if you want to find related proteins or related sequences in a database. Now, then I met the crew from BioStrand, and they asked two very interesting questions, in my opinion. First question was, if we want to find the related sequences, is it really necessary to perform a sequence alignment? And that is an interesting question if you think about that. So maybe in such a lengthy sequence, there might exist a unique barcode, a unique and informative barcode that we could use to relate or to link related sequences to each other. So if you could use that barcode and recognize that barcode into another sequence, and these sequences are also related or homologous, well, that is interesting, but then I can use that unique barcode, index that barcode on the database, and retrieve the sequence very quickly. So I think that's a very interesting idea. And having said that, we already know that concept in computational biology. We call that patterns. And another concept very related to patterns are profiles. So to understand what's going on here, I quickly need to provide you with a short definition about profiles. Typically, profiles are quantitative descriptions. They contain probabilities about which letter sits where in the sequence. And it typically characterizes a protein family, a domain, and it covers very conserved as divergent sequence regions. So typically profiles are long, right? And if you want to align, well, find profiles, you need to become error tolerant. It's different with patterns. The patterns are the barcodes, right? So they are a qualitative description of which sequences, a follow of sequences we should expect. And they're confined to a very small region of 6 to 9 nucleotides or amino acids in a very conserved way. And well, in computational biology, we know such pattern databases. For example, we know ProSite patterns, or we know the Prince database. And these are very valuable patterns. But unfortunately, if you look a little bit closer at these two images I copied from the Interpro data repository, you see there are only a few thousands of patterns available. And that is, well, you could say, let's index that now, and that might be a good idea, but I can already guarantee you that it's not enough to cover with a sufficient density all the sequences that are in the entire ecosystem of biology. It will not work here. So that's where I discovered the second question that the people from BioStrand asked themselves, and that is, can we use techniques from artificial intelligence to mine the entire biological sequence biosphere to find these informative barcodes? And what they did is they applied natural language processing techniques to discover frequent and informative patterns or barcodes, and they are called HIF patterns. And these HIF patterns, there are many of them. At the moment, they're discovered they're still discovered new HIF patterns because mining is still going on. And these HIF patterns, well, they fulfill the same properties as the patterns I just mentioned. They are very conserved and are short tamers that can be indexed on databases. So you can understand here that we move here slowly from a paradigm that goes from the dynamic alignment, maximizing the similarity of sequences, to indexing and the more static algorithm that we're used to in classical database technology. Good. So I was the first to play around a little bit with these the retrieve and relate algorithm, and we set up a benchmarking, and I want to explain you in a few minutes how this benchmarking is executed. So first of all, Ingrid already mentioned, we did a small benchmarking study. We wanted to have a ground truth. So it's not the idea here that we focus on performance measures, like how fast it is. We were planning to set up other benchmarking studies for that purpose. First thing we want to evaluate is like, how well is it to retrieve these homologies that we're looking for, and how does it compare with state-of-the-art algorithms such as blast? Okay. So for this purpose, we use the PDB, a protein database that contains, well, it's a small database, but it still contains more than 500,000 entries of proteins. And the interesting thing is that it contains a three-dimensional shape of proteins. So if we look closer to such a protein, then we see there are some nuts and bolts that sometimes reappear in such a protein structure. And these are called domains. And these domains, they are actually investigated and curated by humans and clustered into a separate database called the CAT database. So CAT, and I don't, I will not explain what CAT stands for, but the important letter to remember is the H here at the end, and that is the category of homologous superfamilies. So all these domains I just mentioned, so this protein is cut up in specific domains, and these domains are then clustered into superfamilies, homologous superfamilies, because they are thought to share common ancestors. So that's exactly the homologous I was talking about earlier. So we have here our ground truth label. So what we will do is we will search, we'll use the protein database as to search. So it contains more than 500,000 IDs and 120,000 unique sequences only. So there are some duplicates here, you can already tell. And the CAT domains we will use as the ground truth query that we will use to search our protein database. Okay, and as you can see, we use the latest daily snapshot of the CAT database containing over 6,000 superfamilies composed out of more than 400,000 domains and so forth. And we see already that not all protein IDs in the PDB are included in the CAT. So we had to do something and build in some sanity checks here to make the PDB look like the CAT database. And I will not go into too much detail, but I will show a slide anyway. So what we did is, and I can answer questions later on, but to cut a long story short, what we did is by post filtering, we made the CAT IDs be present. We mapped the CAT IDs on the PDB and vice versa, because to our surprise, there were some CAT domains that actually ought to come from PDB that were actually not any longer in the PDB database. So after an update, more than 27 CAT domains were removed from PDB. So we mirrored these two databases, and then we used the protein families to query the protein database. Okay, so I will ignore that and focus a little bit about how this querying exactly works. So we considered the 100 biggest CAT superfamilies, okay? And for every CAT superfamily composed out of like a number of sequences and IDs, we randomly selected one sequence and we queried that sequence on our protein database. So remember that all these CAT IDs could be mapped on the protein IDs in the PDB and vice versa. So as a result, this query will give us a result list. Now, let me focus a little bit more on this query here. What is the search algorithm that we use? Well, first of all, we use the BioStrand algorithm via our Python application programming interface. And we had a local install on BLAST with the default setting. So that's why we could not compare the speed, because BLAST was just run locally on an ordinary laptop. But that's okay. We just wanted to compare the sensitivity and specificity. So how did we do that? So bear with me for a second, now it becomes a little bit technical. Well, from this result list, we can see that some of the sequences from the query indeed also sit in our GAT superfamily. So we call these sequences that appear in both lists, we call these true positives. So the BioStrand or the BLAST algorithm rightfully extracted these sequences from our protein database. But there are also false positives. So in the result list, sometimes there are sequences that are not a part of this homologous superfamily. So these are denoted the false positives. So we say that they are related, but they are not, according to the GAT superfamily. And then there are sequences that are, according to the GAT superfamily, related, but we could not retrieve them from the PDB. So these are called false negatives. And then everything else that is not in both, that's outside these two lists, that we denote the true negatives. Okay, so let's look at some results. Well, but before we can do that, I must mention one more thing. There is something in this result list that's also present, and that's called a score metric. So any algorithm will provide a metric that you can use to rank the results. In BLAST, for example, this will be the E value or the P value associated with the local alignment. For BioStrand, this will be the number of common HIF patterns. But this, for the purpose of this comparison, we run local alignment on the client side using a Smith-Waterman algorithm and used that value to rank. But it didn't change much. But it's a little bit more fine-grained result for our curves that we will produce. So we will produce a precision recall curve, and how we do that is we sweep over the score metric, and we will ask ourselves every time the question, like, if I would threshold the score metric at a certain position, how many true positives, false positives, and false negatives do I have? And I compute precision. So for those that do not know how to compute precision and a recall, it is the ratio, the proportion of true positives on the true positives plus the false positives, so precision. And the recall is the true positives, the proportion of the true positives on the true positives plus the false negatives. And you can see immediately from this curve, so on this curve we have precision on the y-axis and recall on the x-axis. The yellow curve is the curve produced by the BioStrand algorithm, sweeping over this local alignment score. The blue line is the BLAST algorithm. And you can immediately see that the problem that we addressed here is extremely different. BioStrand cuts off a recall of 30. So it means that from our 100 repetitions, repeated searches, only 30% of the true positives that were truly in the list could be recovered. So it is a complicated problem, and it's complicated because the GAT database focuses on structural homologies, and structural homologies are not necessarily the same as a sequential homology. So they can differ quite a lot. So that was why BLAST and BioStrand had difficulties to come up with a large recall figures here. And you can see another thing, you can see that BLAST cuts off here at a certain precision, and that the BioStrand drops. There's a good reason for that. The BioStrand algorithm currently does not include a reporting threshold, or does not include a filtering of the false positives. And that idea is a little bit borrowed from Google PageRank, where you actually give the end user a ranking of results. rather than saying like, okay, you cannot click to the 5th or 6th page of your Google search if you want to. So instead of a top result, the BioStrand algorithm will report everything that contains the HIFT patterns. Okay, and the ranking is actually based on the number of HIFT patterns. So that's why we see also precision will drop to this value here. Another thing that you can see in this curve is that there is indeed a 10% difference in what could be retrieved. And the reason for that is, as I mentioned before, it is a very complicated problem, and BLAST will do is a dynamic algorithm. It will optimize similarity between sequences, whereas the BioStrand algorithm is a static index-based retrieval. When you have a complicated problem, then percentage identity of sequence similarity is very low, and it is difficult, obviously, that to retrieve all the sequences. And we can see that actually in the next slide. You can see that better in the next slide. So here I provide a histogram plot. Let me quickly explain these two plots. So the first panel, the top panel, are the true positives. The bottom panel are the false positives. And what is in this plot is it's not a stacked histogram. Sorry, it is a stacked histogram, and it's stacked according as follows. So you see, BLAST is the green, BioStrand is red, and what they find in common, the true positives they find in common, is denoted by blue. Okay, and what I did is I count the true positives, obviously, but sorted them according to the percentage identity. And interestingly to see here is that both BioStrand, the fast indexing approach, static algorithm, as the dynamic BLAST algorithm, were able to retrieve most of the high percentage identity sequences. It's only when percentage identity drops below, let's say, 40%, that the dynamic algorithm improves and is able to also recover these sequences which contain a lot of dimensions. Okay. Another interesting thing to see is that if you look at the false positives, well, there are no false positives that have a high percentage identity. So the percentage identity of the false positives included by BioStrand, and yes, there are a lot, but do note that there's no reporting threshold applied here. They all have a very, very low percentage identity. And I guess this graph will be a very interesting way to think about how false positive filtering could be executed in the future. Now, the question I often receive is that, like, if you are an end user, is this trade-off that you have here between sensitivity and speed, is that important? And well, I'm not an end user, I'm an academic person, so it's hard for me to tell. But what I can tell you is that, for example, there is a different, there is a finer granularity in the CUT database. It's called CUT-S. So that means that there is an additional category within the superfamily that clusters are categorize the sequences that have a sequence similarity larger than 5%. If we would have used that category, then what would happen is that the whole left bunch of counts will just move to the false positive. That would not look good for the BLAST algorithm either. So the real question is the trade-off, is that important to an end user? Well, the real answer to that question is that it depends on the application. For example, if I was an evolutionary biologist and I want to find very related but divergent sequences, well, then I might want to go with a slower algorithm and try to relate these very divergent sequences. But if I'm now like a drug developer and I work on model organisms and modern organisms, right? And I want to interlink a lot of types of different databases and speed is an issue, well, then I might want to go with the biostrand option here to retrieve that, because percentage identity is of these sequences between modern and model organisms is usually high. Now, another question I receive is, does it matter if a search takes 5 minutes or takes 15 minutes? Well, I cannot value that. I don't know what is the price for an employee sitting idle for 15 minutes. But what I can tell you is the following. So I'm an electrotechnical engineer as well, and I studied a lot about computers and computer architectures. And when I was still at the school banks, I learned about Moore's law. And well, Moore's law said that computer power would double every 5 years or 3 years. But the bad news here is that that was true when I was sitting on high school, but today, Moore's law is pretty much that. So we cannot expect much improvement anymore from the silicium technology that we have. Expect from scaling up. So we can make a computer twice as fast if we buy 2 computers. Okay, so so we cannot miniaturize much more, and there's not much more speed gain from the old silicium technology. On the other hand, you have the Carlson's curve, and well, and I think Carlson's curve can also be called a law. And here you see the cost to sequence a human genome in US dollars, and you see that from 2000 on, it significantly drops, and here at this point, even it outperforms Moore's law. So I expect actually the cost to sequence genomes will even drop. And if it doesn't drop, it is also a scalable thing. So to sequence faster and cheaper, we can double. So what we are looking at now is that there is some type of arms race between silicium to do sequence alignments and the sequencing technology that is scaling up and causes databases to almost overflow. So how to resolve this issue? Well, there is only one way that we might expect, and that is transition to another technology group. So maybe we need to go to quantum computers or more biological inspired computers and transition to another S-curve, as we say. But I don't think that it's going to happen in the near future. Another way that we can do is simply move from the dynamic algorithms that we have, from the blast-like algorithms to more static algorithms for biological sequence retrieval. And this is to me a valid solution for a very complicated problem. And by that, I would like to conclude my talk. Thank you for the questions, of course.
Thank you very much, Dirk, for your very clear presentation of this really difficult topic. So you really explained it very well. Thank you for that. We will move on to the next speaker, Joris Van Houtven. Joris will talk about a topic that was a part of his PhD thesis, and it's about Prism, assumption-free pattern matching in mass spectrometry-based proteomics. Please, Joris, go ahead.
Thanks, Ingrid. I'll stop Dirk's presentation and start mine. Here we go. Can you see that clearly?
Yes.
All right. So Prism, that was a workflow in proteomics that was a significant part of my PhD. And I'll be explaining to you how it does pattern matching in mass spectra using the CPC algorithm. That's an algorithm we developed specifically for that purpose. I'll start with why we are interested in proteins and proteomics and what it means to do that with mass spectrometers. And then I'll tell you how we identify those proteins using Prism and CPC. And finally, I'll have a short discussion about what that means in a broader context. What can you do with this technology? Good. So why proteins? Well, proteins, after water and fat, they make up the majority of your body. They make up your muscles, your nails, your hair. They transport oxygen in your blood. They do everything. They are the tools of our bodies. So they're very important. And what do they look like? Well, here's a picture and the corresponding protein code as well. So I want to zoom in on that a little to find out what that is all about. And proteins exist out of amino acids. Like these are even smaller molecules. For example, here is alanine, which is denoted by A, and you can find this A back in the protein code. And these amino acids, there are 20 of them, as you might know, they are linked together like a chain to form the different types of proteins. And the point here is that the sequence, the order of those amino acids determines the function of the protein. So that's important. Now, what do we do with those proteins? Well, we want to find biomarkers. That's why we're interested in those proteins. And we do that in a differential expression analysis. So let me unpack those things for you right now. Here are two pictures. One is a healthy piece of liver tissue, and the other is a piece of liver tissue with cirrhosis. What we now want to do is investigate the proteomes of those biological samples, meaning we want to find out which proteins are present and how many of each type. So for example, this one could be overexpressed in the liver with cirrhosis, and another type of protein could be underexpressed, and then many other types of proteins could not be differentially expressed. There's not really a difference. Now, of course, it's these top two that will be of interest to us, right? Those might be biomarkers. Biomarkers are interesting in doing medical research as well as in the pharma sector, because those might be the targets for inventing new types of drugs to combat, for example, in this case, liver cirrhosis. How do we do this in practice? Well, we use mass spectrometers. Those are high-tech instruments, which are basically expensive scales for very tiny things like proteins. And they have other application domains as well. And what we do is extract proteins from a biological tissue and put them all together into the mass spectrometer. And this instrument will then break them up and analyze them, and will spit out many of those mass spectra, those graphs here. And now each spectrum represents a piece of protein, like a chunk. And it's our task to then find out what protein this chunk belongs to. Now these bad boys, they cost about let's say €500,000 on that order of magnitude, and they spit out about 20,000 spectra or even more these days per hour. So that's a lot of data to go through. And that's good because we want to cover as much data as possible to find our biomarkers, but only 50% of these spectra get identified. So now is the question, what about this other half of spectra? Maybe my biomarker is in that other half and I really want to know what's in there. Maybe this other half does not contain a lot of information, or maybe the conventional identification methods just don't see what's inside. They have blind spots. And that's where I would put my money. Because if you look at this Venn diagram, this is one data set processed with 3 different identification methods. And they give 3 times a different result, right? So about 2,000 identifications are the same, but many others are different. And why is this happening? It's because each of those methods is based on their own conception, their own idea of how one should do identification. And that means they all use their own methodologies and their own heuristics. Heuristics are let's say, shortcuts to speed up the calculations, but unfortunately at the cost of sensitivity. And that's precisely where they introduce those blind spots. It's the ambition of Prism then to become the outer Venn diagram, which is able to identify everything that is identifiable. And we do that by looking at everything there is to look at, doing that very thoroughly, and then we will be applicable to all types of data. We can do that irrespective of the data type, which is not always true for some of those other methods. Okay, now how do we do that specifically? Well, before I get to that, let me just quickly reiterate that such a mass spectrum is a chunk of protein. It's like the fingerprint of a protein. If you're an expert, you can actually interpret this by looking at the peaks, the locations, or the distances between the peaks, and infer the protein code from there. And that's exactly what some of those identification methods will do. Typically, they will take this observed spectrum and query it against a database of all theoretically possible calculated spectra. It's like you have a fingerprint and you look it up in a database of all known fingerprints, something like that. But with Prism, this database is actually consists of patterns. And patterns, we then, so how do we match then a pattern with an observed spectrum? That's why we have our CPC algorithm. It's like the mathematical convolution operators, but in a discrete form. So let me show that to you. We have a pattern from our database here. And here is an observed spectrum. And what we will now do is write down for each possible alignment of these two. So for example, here we have an alignment of just two of their peaks. We will write down the score. So we have only one pair of peaks aligning here. And we will build up a score trace by now sliding. So keep an eye on this. We now slide across, each time matching at least one pair of peaks, until at some point we have a pattern match. And then, of course, our score trace will be very peaked. And we do that across all proteins, across the entire patterns, and we end up with something like this for each protein. So that's what the score trace then looks like. And Prism uses its statistical machinery to pick a winner from all of the output from the CPC algorithm. And this winner, this best matching alignment, then of course points to the correct protein. Good, but please do know that we do this across many score traces because there's one of these for each protein in your database. And that's a lot of data. That is a lot of calculations. And that's exactly one of the reasons that we are doing this today. And that's because back in the 90s, people simply did not have the computation power to do this, right? Because if you think about it, it's a convolution. That sounds quite simple, and it actually is. Prism is the methodology behind Prism is relatively simple, but the computations needed back then were just infeasible to do. So they had to use their shortcuts, they had to use heuristics, and they had to introduce, unfortunately, blind spots. But today, this is a new era, and we don't have to do that anymore. So what does that mean from a broader perspective? Well, first of all, you don't have to make any more assumptions about your data, and your software does not need to be restricted to specific parts of the search space. And you can make your application truly universal. And that's because all of those ingredients, those heuristics, those shortcuts that people usually used when they made a protein identification method, they aren't necessary anymore. We don't need to think about acquisition methods or worry about enzyme specificity. Prism can do it, can leave it all behind. Now, does it work? Have we tried it? Yes, we have tried it. So we took 100 spectra that were easily identifiable with Sequest. That's a well-established search engine. And it turns out Prism can agree with 84 of those. Now that sounds good, but not great, right? 84%. But actually, these last 16, we looked at them manually, and we know what's going on there. We know why this is not in agreement, and it's actually relatively easy to make updates to Prism and then fix those as well. So we can get very close to complete agreement with Sequest on those top 100 spectra. Okay, that's interesting, but that's not what we were looking for, right? We wanted to cover up those blind spots. We want to find more than people were able to find at this moment. So we also ran Prism on 100 spectra that Sequest could not identify. And in 15 of those cases, we could already find significant results. I've shown them here. These are portions of protein code. And these 5 red ones over here all belong to the same protein, this protein right here. And it's interesting, when I went to investigate, it turns out that Sequest would actually expect this protein chunk to show up, but the first portion was not visible. So that was unexpected, right? And that's probably why Sequest did not pick it up. But for Prism, this doesn't matter. Prism can just identify it anyway. It can identify patterns or portions of patterns. So when Dirk mentioned mutations earlier, this is one area where Prism could be of use as well. But I think at this point, as the project is now, we could first think about using it to provide a universal benchmark. So to set a gold standard for identification, regardless of the data type that you are using. So you can properly start comparing methods with each other. Because up to this point, as I've shown you in those Venn diagrams, existing methods come up with some plausible match, like a plausible identification for a spectrum, but who knows, maybe it's wrong, right? Maybe it's actually another protein that fits the spectrum better. But with Prism, you check everything, right? So we know that this should be the optimal match. And that's how we could set this gold standard. And using that standard, we can improve other methods as well, right? Because now, when you develop a very fast identification algorithm that is industry worthy, let's say, you in the past you were never really sure when you use a heuristic or when you use a shortcut to be fast enough, you were never really sure what its impact would be on the quality of your output. You know that it's faster, but what about the sensitivity? And now we can measure that accurately with a gold standard. So that's a boost to anything that is involved with proteomics, any research or medical applications as well. I could talk a bit about how we could easily make Prism more statistically powerful and faster, and how that leads to even more applications. But in the interest of time, I'll just highlight one, and that's the detection of post-translational modifications. That's still early work, but I have seen that in these core traces is information to tell you without prior knowledge, which modifications might be present on your proteins. And that's of very significant biological interest. That would be a big milestone if we were ever to achieve that. And lastly, I'd just like to say how Prism relates to machine learning, because everybody's doing machine learning nowadays. Many identification methods use it, and they're quite successful. So what they do is they have a training data set of identifications that they know. They train their machine learning method. That's a supervised training. And then you have a very good identification method. Okay, but what if you want to identify spectra, proteins that you've never seen before? Well, then they say we could first do some unsupervised machine learning to find structures in spectra that we are not able to identify. You could flag that. You could flag those spectra, but in the end, if you want to identify something that you've never seen before, then you need to use a method like Prism to actually be able to reliably label those new observations. So whatever the future holds, I think a pattern matching approach like Prism will always stay relevant. And with that, I'm happy to take any questions.
Thank you very much, Joris, for your very clear presentation. And yeah, it's a very difficult topic again, but yeah, you really take us into the story, explain it very well, and show us all the wonders that those patterns can bring about and can reveal. So thank you very much for that. I think for the interest of time, we have time for 1 or 2 questions.
Yeah, maybe I will start with Joris because I know he has some time restriction, let's say. So we have one question about where you would see the most value of your Prism algorithm or application in regards to the discovery or identification of new protein, like synthetic, in the field of synthetic protein or in the field of organically produced proteins.
Okay, so everything I've discussed up to this point is from the assumption that we are looking at organic proteins, and even for organic proteins, we've seen that they don't all follow the same rules of fragmentation. They don't always produce the spectra that we expect them to produce, right? That's why Sequest probably missed that one protein that I've highlighted. And that's where Prism could surely make a difference, right? So for sure in organic proteins, it can make a difference. And if you then start making synthetic proteins, which may or may not produce even more exotic spectra, you can actually make them more exotic. And as you make them more exotic, existing methods might become less and less able to easily identify them. But I would look at it this way. If a tool like Prism is available to you, that you don't need to worry about the detectability of your synthetic proteins, because for Prism, it does not matter. So I think in both domains, it can have an added value.
Thank you. And maybe for Dirk, I think you have shown the nice validation of this of retrieve and relate search engine, and what do you think could be improved for the next step, because you have seen it with your bioinformatician eyes?
Yes. So I think the results I showed today are very, very interesting and convincing. I believe that the static algorithms are ahead a nice future actually. So, but what you could see from the precision recall curve is that with BLAST that has a type of score metric associated, or let's say a p-value associated with each sequence that it gives you back. That is not the case with BioStrand. So the move from the PageRank ID, which is very, very interesting, obviously, and maybe a lot of users wanna have that because they want to click to the page number 100 to see if something interesting could be found there as well. But give the users an option to, for example, try to trim the false positives a little bit, and come up with a new score metric to come up with this ranking of and filtering of the false positives. Now, showing the percentage identity plot on one of my last slides, I think there is much material for discussion on how to tackle and construct a scoring scheme to do this false positive filtering. But I think that is an additional feature to, an interesting feature to have for users that do not want to retrieve 100-page results, for example. And maybe they want to just have the top 20 significant hits.
Yes, I think indeed, and I think the showing the metrics, the percent identity, that is something that we definitely are planning to do in very short time. And to come back also on the identity scores, our first focus was to reach a score of, let's say, 40–50%, because then you have a very nice trade-off between the scalability, the speed, and all the advantages that you want to have versus accuracy. And now we can look into the next step and go into the low identity scores. And that's something we definitely have ideas about. And that's also something that we are going to address in a different way. So yeah. So by this, I want to end the webinar. I really thank you all for attending. I just want to point out, next month we are there again. Then we have Professor Joy Arobi of the Biomedical Research Institute of the University of Hasselt. We have also a collaboration with her, and she will talk about extracellular vesicles in multiple sclerosis. So I hope to meet you again next month. Thank you very much. Bye-bye.